Okay. My name's Travis Craddock, and I come from Nova Southeastern University in Fort Lauderdale, Florida, which is in the southeast of the state, right down by the tip by Miami. This is our campus here, and I work here as director of the Computational Medicine Group and Clinical Systems Biology in the Institute for Neuroimmune Medicine. And we're a large conglomerate that deals with clinical scientists, laboratory scientists, animal scientists, cellular scientists, and in our group, which is the computational team. And our whole purpose is to try and find novel treatments and diagnostics for complex chronic neuroinflammatory diseases. And this involves, classic neurodegenerative diseases, but in addition, diseases such as myalgic encephalomyelitis, which is also known as chronic fatigue syndrome, Gulf War syndrome, as well as most recently long COVID. I just want to put a shout out to my team here. This is a growing team. We kind of shrank over the pandemic, but we've managed to rebound quite well. We've got two new faculty in the group, Dr. Kostas Sidropoulos and Dr. David Casada, and then several staff and students who have been working with us. Today, in specific, on the left-hand side there, you'll see Isadora Dabro, and a lot of the work that I'm going to be discussing today is done by her in the lab. I'd also like to thank our funders, various organizations, the NIH, the DoD, Nova Southeastern University, and our computational platform, which is hosted by the University of Miami down in Miami, Florida. And finally, I'd like to thank the organizers of the conference for inviting me and having me come here to speak, because it's a fantastic city. Never been to Sicily before, and I find it quite amazing, especially the fact that there was almost a volcanic eruption when I was here. All right, so just a quick overview of what I'm going to be talking about. A lot of definitions here. I'm going to be talking about what are psychedelics, giving a gross overview of that. What is the microtubule cytoskeleton? A lot of you here probably know what that is, but then again, some of you may not. So I want to go into the details of that. And then I want to talk specifically about the most recent work that we've been doing and how do psychedelics, or a specific class of psychedelics called phenylethylamines, how do they interact with the microtubule cytoskeleton? Then I'm going to switch sides and go from psychedelics, which are believed to expand consciousness, to the other end of the spectrum and talk about general anesthetics, which are believed to erase or suppress consciousness. And talk about some older work that I've done with Stuart and others like Philip Kurian, and talk about general anesthetics and how those interact with the cytoskeleton, and then discuss the implications of these findings for a theory that was put forward by Stuart Hameroff on the quantum mobility theory of anesthesia and consciousness. And I'll conclude. Okay, psychedelics. What are psychedelics? They're a subclass of drugs that are known as hallucinogens, and they're any sort of psychoactive chemical that gives the imbiber or the victim, and depending on the case, some sort of non-ordinary mental state, which is usually referred to as a psychedelic experience or a trip. And what it is a change in perception, usually auditory and visual changes in perception. Because of this, because it's a change in the way that you view the world, it's viewed as one of the natural probes of consciousness, with the other being general anesthetics, which, as we know, turn off consciousness and allow for great things in medicine like surgery. More so these days, we're starting to learn about how psychedelics may be used in treatments of illnesses, especially things like depression, post-traumatic stress disorder, and so forth. But I'm not going to be discussing those aspects today. Now, the classic psychedelics, there's three main classes of those. There's tryptamines, lysergamides, and phenylethylamines. I'm going to be talking today specifically about phenylethylamines. But in general, these classic psychedelics are all believed to exert their influence through the serotonin receptor family, specifically a receptor called 5-HT2A, which is a specific serotonin receptor. However, that is the most prime target because it's been shown that these chemicals strongly bind. It doesn't necessarily mean that that's the only place that they act. There are many other receptors and many other targets where these drugs act, which may either be the prime source or it may be something that gives rise to the richness of the experience and the differences that you experience in these sort of psychedelic trips. So what are phenylethylamines? So phenylethylamines are a natural class. So first off, a phenylethylamine is a chemical structure which is made up of a benzene ring double bonded to a amide group at the end. And this alkaloid can be modified in many ways to give rise to many different structures. But that common backbone is the main structure of a phenylethylamine. So if you modify it, you can get your standard neurotransmitters like dopamine and norepinephrine, or you can get things that are known as non-psychedelics, non-psychoactives, like albuterol and phenylephrine. Now, besides the point, I have albuterol. I use it for asthma on occasion. However, I was doing some research on this, and I was like, oh, it's non-psychoactive. However, as I was looking through the literature, I found out that it can be psychoactive. There has been some cases. It's not known whether it's due to that drug or if it's due to the propellant. I just want to raise that at certain doses or certain situations, these may have other effects. These are just broad categories that I'm stating here. Psychostimulant effects, primarily the one that is known for illicit use is methamphetamine, or meth, or if you've ever watched the show Breaking Bad, that's the drug that they sell in that. And then the classic psychedelics like mescaline, MDMA. And these are known drugs that are used recreationally to give a high, give a psychedelic experience. They all have very high affinity for the serotonin receptor, but again, they can target other receptors in the body, such as adrenergic receptors, dopaminergic receptors, histaminergic, opioid receptors, and more recently, they've discovered things like trace amide receptors, which are specific to some of these not as prevalent phenylethylamines that are naturally produced in the body. Now, these exogenous chemicals, the ones that are imbibed from external, are believed to act on these same receptors that natural phenylethylamines would target. Now, looking at that structure, the phenylethylamine structure, it was discovered back in the 1990s, or wasn't discovered, it was realized back in the early 90s by Van Wokerum, that these structures very closely resemble drugs that target the microtubule cytoskeleton. So if you're looking, I don't know if I have a pointer up here. I don't think so. But over there on the bottom left there, that's 2-phenylethylamine. Okay, so that's the basic structure of phenylethylamine. And if you look up at colchicine, which is a known microtubule destabilizing agent, on the bottom right, you'll see that ring structure with the 3 O's attached to it. And then following up the right, you see that chain where it goes up to the NH group. And that is the structure of the phenylethylamine. And everything else on that is a modification. You can see that same structure in mescaline over in the bottom right-hand corner, where it's got the 3 methyl groups with the methoxy groups with the oxygens there, going up to that nitrogen group up at the top. So he was noticing that this similarity between mescaline and colchicine, and then he generally classified this as colchicine is related to phenylethylamines. He did the same thing with tryptamines, and if you look at tryptamines, vinca alkaloids very closely, which are other microtubule destabilizing agents, they share a common backbone with tryptamine hallucinogens as well. So he put forth this idea, do microtubule targeting agents, could they be related to these psychedelic agents? Now, there was some work done way back when, back in the early to mid-70s, on mescaline binding to tubulin, and it was shown that mescaline does indeed bind to tubulin, and that it inhibits microtubule formation. So tubulin is the protein that makes up microtubules, and when bound to mescaline, mescaline stops these microtubules from forming. Conversely, there's more recent work, very small amount of literature back in the 90s, where it's looking at 2-phenylethylamine on the bottom left there, and how it binds to microtubules, but instead of destabilizing it, it stabilizes it. And so it gives rise to this idea of what's going on here. Do phenylethylamines destabilize? Do they stabilize? It's obviously going to depend on the chemical structures that are involved. But it does suggest that there may be a potential neuromodulation of neuron function via the cytoskeleton through phenylethylamines acting directly on the cytoskeleton. And so this is where I come in. So moving over to the cytoskeleton, what is the cytoskeleton if you don't know? So we're talking about consciousness. We're all coming to this idea that, maybe not all, but a large proportion of us would like to believe that the consciousness arises from brain activity, and your brain is a collection of nerve cells and brain cells known as neurons. Now these brain cells are very highly structured. So you have a cell body, like over here on the left, you have this cell body and these little extensions off of it called dendrites, and this very long extension called an axon. And the neuron receives signals coming from the dendrites, so other neurons will connect to the dendrites and give signals to the neuron. That information is integrated in some way, and it decides whether it's going to fire an action potential or not. And then it sends that signal, if it decides to fire, down the length of an axon, where that axon then connects to other neurons, and so forth. Now, if you look inside of a neuron, it's very highly ordered, and this is primarily due to the orientation and structure of the microtubule cytoskeleton. So the cytoskeleton, as the name implies, gives rise to the shape and structure, the skeleton of the cell. And within a neuron, you get these bundles of microtubules that form parallel bundles going down the axon, anti-parallel bundles in dendrites. Now, when we undergo things like learning and memory, there's reorientation of this microtubule cytoskeleton within dendrites and within the axonal growth cone in order to change the synaptic strength between a presynaptic and a postsynaptic neuron. And that's characterized in the long-term potentiation model of learning and memory. It's also been shown that a large host of neuropsychological, neuromuscular, as well as neurodegenerative diseases have not at their base, but in their pathology, they have dysfunctional microtubule cytoskeleton. So work by Avner Priel, Jack Tazinski, and Nancy Wolf back in the early 2000s. They put out a book called The Nanoneuroscience, Nanoneuroscience, and there's a chapter in there specifically talking about how illnesses such as Alzheimer's, Parkinson's, Huntington's, as well as schizophrenia, bipolar disorder, autism, how all of these have some sort of dysfunctional microtubule cytoskeleton at some point. And so this is a natural place to look for ways that we can restore function to the cell to try and alleviate some of these illnesses. Now, the microtubule cytoskeleton itself is made up of a protein called tubulin, which is over here on the left. It's made up of two components, two almost identical proteins called alpha and beta tubulin. They are constructed separately, but they're never found apart from one another within the cell. They're joined almost immediately. And then they have these things that kind of stick off of them called C-terminal tails, and they're very important for the function of the protein in polymerization. Now, what these tubulin proteins do is they stick end to end, and they form these long strands called protofilaments, and then the protofilaments stick side to side, and then wrap around to form a hollow tube, and that hollow tube is what we know as a microtubule. These things are about 25 nanometers in diameter, and they can range in length from, a few, that should say micrometers there, up to millimeters in length, depending on where they're located and in what cell type. It's the most abundant structure of the cytoskeleton, and like I mentioned, anything that affects a microtubule cytoskeleton is going to affect structural and synaptic plasticity. There are a host of drugs that we know that target tubulin, and they are responsible for one of two things. Either they promote stabilization of the microtubule cytoskeleton, or they inhibit stabilization. So there are classes of drugs such as taxane. If you know anyone who has undergone anti-cancer therapy, you may have heard of the Taxol drugs, paclitaxel. That is a common drug which is used to target the microtubule cytoskeleton because the cytoskeleton itself is involved in mitosis, in dividing cells, and because it stabilizes the cytoskeleton, that mitotic spindle cannot collapse, and therefore the cell, whatever cell is trying to divide, like a cancerous cell, will die because it gets stuck in the middle of that process. Other drugs like colchicine and vinca alkaloids, they bind to tubulin and prevent it from forming a microtubule. So you hit it on the other end, so it can't even form things like the microtubule mitotic spindle. It's rare, but there are anecdotal evidence and case reports out there that patients undergoing anti-cancer therapy or treatment for gout, which is what colchicine is primarily used for, that they experience hallucinations. It's attributed to neuropathy in some case, and that makes sense in regards to the fact that these drugs would target the microtubule cytoskeleton, and if you destroy that, then your neurons are gonna suffer. However, it does suggest that alterations in microtubules may play a role in auditory and visual hallucinations. Okay, so coming specifically, what we wanted to do is we wanted to look at phenylethylamines, a class of known hallucinogenic drugs, and we wanted to see how well do they bind to the microtubule cytoskeleton. So we performed a computational study where we took 100 phenylethylamine compounds and that are known to have psychedelic effect and bound them to the colchicine binding site on tubulin, and then ranked all those drugs based on how strong they bind to the cytoskeleton. Can you guys? Oh, you guys can see that. So here's colchicine up at the top, and it binds with a fairly strong binding energy of negative 10.8 kilocalories per mole. And these are the top 10% of the phenylethylamines that bind to the microtubule cytoskeleton. I can't list their names because we're currently patent pending on some of these, so bear with me. They are all phenylethylamines, and there's going to be a publication coming out very soon, but until then, we're bound by copyright at this point. So we rank them based on the binding energy, and these top 10 bind fairly well to tubulin, but not as strong as colchicine. So here we're ranging from -8 to -9 kcal per mole. Now, if we look at the whole 111, they go down to about -5 kcal per mole, which is a weaker binding energy. The closer you are to zero, the farther away you are into the negative, the better it is a binder. And so that's not a horrible binding energy, but it's not the best. But these ones here specifically, show a very strong, moderate to strong affinity for tubulin. Then we wanted to actually look at these experimentally and take some of these predictions and test them out. Can these things bind to tubulin, and can they exert some sort of effect similar to colchicine in this case? And so what we did was we screened them through a software that looks at the pharmacokinetic and pharmacochemical properties of these compounds to look at things like their absorption, their digestion, their excretion, and basically how they're metabolized within a cell. And what we found here is that all of them, as expected, easily cross the blood-brain barrier. Unlike drugs like colchicine and Taxol and standard antimitotics, which find it very, very hard to get into the brain. The phenylethylamines that are causing psychedelic effect, obviously, it makes sense that they would be easily entering the brain. Then we looked at how they are metabolized within a cell. And these are all enzymes that break down drugs. And so we wanted to see which are the ones that most likely would not be metabolized quickly within the cell. And so these three compounds, PEA, phenylethylamine 1, 4, and 6, were only interacting with 2 of these enzymes at most. And therefore, we decided to move ahead with those because once they get into your body, it's more likely that they're going to stick around in their regular form before being excreted into the body. So then we moved ahead and we decided to take these compounds and put them into microtubule polymerization assays to see the effect that they have on microtubule dynamics. Now, 2 of those drugs we had to go through a bunch of scheduling and stuff like that, but we ended up getting 2 of those drugs quite easily, while the third one was very difficult to obtain and was only obtained very recently. So I'm not going to be talking about that PEA1, because it was, we've only had it in the lab for about a month now, even though I ordered it well over a year ago. Anyway, so we performed tubulin polymerization assays on 2 of the 3 compounds, and this is what we find here. So the black curves there are standard control microtubule polymerizations. And so what we're seeing here is what's known as optical density. So as these proteins are sticking together, we're shining light through it and measuring how much of that light is being blocked out by the structures that are being formed. And so as those microtubules are growing, the optical density is going to increase. And so that black curve is the normal curve that you would expect for tubulin polymerization. Standard stabilization is done with paclitaxel, and so you can see that red curve there. It forms much quicker, so it means that it's enhancing the polymerization activity of the microtubules. And then colchicine in the orange there, you can see that it's very, very slow, as well as you're not getting as much microtubules forming in the entire system. And that's because colchicine is preventing microtubules from forming. Now, if you look at the various concentrations of these 2 compounds, PEA6 and PEA4, you'll see that at comparable concentrations to paclitaxel and colchicine, you're not getting much of effect or much difference from your control group. Maybe a slight increase for PEA6, not really much anything for PEA4. But as we ramp this up, remember these don't bind as strong as colchicine, so you'll need a little bit more in the system. As you increase it up, you can see that curve starts to increase. So at 50 micromolar, we're getting comparable between control and paclitaxel for PEA6, almost at the polymerization rate for paclitaxel for PEA4. And then when we move into 100 micromolar, which is fairly strong dose, we get a very, very robust polymerization of the microtubule cytoskeleton. So we are seeing some sort of an effect here. We know that these drugs, we're predicting that they bind to tubulin. We're seeing that they do have a dose-dependent effect on microtubule polymerization. However, let me step back. We know that they are affecting tubulin polymerization, but we don't know if this is actually forming microtubules at this point. We know that it's increasing tubulin polymerization into some sort of object, but we don't know whether it's tubes or clumps or some sort of other aberrant form like sheets. Tubulin's known to form under various physiological conditions. So we decided, okay, we better look at this and look at under the microscope to see what's actually being formed. And that's what we have here. So on the upper left there, we have our control group. Then we have paclitaxel, and you can see these lines here that are forming. And these are the microtubules themselves. These are our preliminary results. We're trying to deal with the concentration of tubulin right now. We believe it's quite high in these systems, and that's why it's so, you get these kind of swirl patterns that are forming there. It happens to be too much tubulin there. However, the thing that to note is that the structures that are being formed are these lines here, and these lines themselves are microtubules of the same, the same scale that we would expect for microtubules. So in the case of PEA6 and PEA4, if they were forming some sort of aberrant formations, what we'd expect is more maybe larger chunks of what's being observed there over in colchicine. You see colchicine, you get these little aggregates, but no lines being formed. However, because PEA6 and PEA4 are forming the filaments of microtubules, we can say with certainty that most likely these things are contributing to microtubule polymerization as opposed to some other aberrant formation of microtubules. Okay, moving on. Going to the other end of the spectrum, what is a general anesthetic? General anesthetic is anything that blocks conscious sensation of pain, prevents memory formation, paralyzes you, and produces unconsciousness. However, that point of hypnosis, I believe, has been removed from recent definitions. They say that it doesn't have to produce unconsciousness. As long as it does the other three, you really can't tell the difference if it's producing unconsciousness or not. You're not going to remember it, you're not going to feel it, and you're not going to move when it happens, which is kind of a scary thought to think of, that you may actually be experiencing your surgery and just not remember it afterwards. I don't think that's the case. I think that hypnosis is a very important point. However, we'll leave that for other people to talk about. These are required for all major surgeries. The things that are known about anesthetics is that their potency correlates with their solubility in lipids, so their solubility in oil. The more soluble it is, the more potent it is. It's believed in the larger literature that the prime site of action is membrane receptors. However, this has been argued against because there's various anesthetics which have differing effects on the same sort of receptor. So, for example, GABA is a prime receptor, and it's my understanding that certain anesthetics will cause an increase in GABA receptor current, and while others will decrease it, depending on which class of anesthetic you're looking at. However, it's also known that general anesthetics target the microtubule cytoskeleton. So this was originally suggested by Claude Bernard in 1875. It's also known that volatile anesthetics at very high concentrations cause aberrant formations of microtubules. And then more recently, in the last couple decades, Rod Eckentoff's group at University of Pennsylvania has done a lot of work that has shown interaction of anesthetics with microtubules. So, for example, desflurane, which is a volatile anesthetic, changes tubulin expression up to 3 days after exposure. It also changes expression, sevoflurane also changes expression of tubulin out to 28 days after exposure. It's known that halothane binds specifically to tubulin in humans. It's also known that anthracene, which is a fluorescent anesthetic, also binds directly to tubulin. It's known that patients undergoing taxane or Taxol treatment for cancer, remember Taxol stabilizes microtubules, is that they require a higher dose of anesthesia in order to experience the same effects as someone who's not taking a microtubule stabilizing agent. And then additionally, of 500 detectable proteins within, this is in a rat brain, 500 detectable proteins, tubulin was among the 2% that was affected by halothane, the 1% that was affected by isoflurane, and one of 3 protein targets that was affected by both of these anesthetics. The other ones I believe were a heat shock protein and I always forget what the last one is. So look up the reference. I'll give it to you at the end. So that's great. Okay, so we have these targets, membrane receptors, we have microtubule cytoskeleton as potential targets for general anesthetics, but what do they do? How do they act? It's known that anesthetics use weak van der Waals forces to bind. They're not covalently binding, they're not binding very strong. On that scale I was talking about earlier, they're binding at about -2 kcal per mole, -3 kcal per mole to a target. So it's very, very weakly bound, and it's mediated by these London dispersion forces. And therefore, it's going to rely on things like its hydrophobicity, its solubility in lipids. So the more soluble it is, the more likely it's going to get into these protein hydrophobic pockets. As well is it's related to its permanent dipole strength. So there's, depending on the permanent dipole of the anesthetic, that's going to affect the anesthetic action, as well as its polarizability. And I'll show you a slide in a minute what I mean by that. Now, this led Stuart Hammerhoff back in '98 to put forward a paper talking about how anesthetics may potentially work by binding in microtubules, and this eventually became known in the literature as quantum mobility theory of anesthesia. And in this idea is that within microtubules, you have tubulin, which forms these lattices, and tubulin has this large amount of aromatic amino acids, which are tryptophan, tyrosine, and phenylethylamine. And this is exactly where anesthetics like to bind. And so the idea is that these aromatic amino acids, which are highly polarizable, can set up oscillating London dispersion forces, and they normally oscillate back and forth. However, if you take an anesthetic and you put it near one of these, is that it would inhibit that oscillation and therefore destroy consciousness via the Penrose-Hammerhoff or mechanism to generate consciousness. So this is the idea, is that anesthetics would suppress these London dispersion oscillations. Back in 2017 or so, myself and Philip Kurian, we did some work to try and test this and say, okay, that's a great theory. Let's actually put it to the test and measure, is that something that could actually happen? So if we look at a group of volatile anesthetics, which are labeled in the light blue there, and compare it to chemicals which are known as non-anesthetics, and put it on this graph of solubility in olive oil compared to its potency, the minimum alveolar concentration required to induce anesthesia, we get this nice graph here, this nice trend line, and this is known as the Meyer-Overton curve. And so this predicts the strength of the potency of an anesthetic. However, it also predicts that these other compounds, like the two in red there, which I believe are F6 and trifluoromethylbenzene, that they also have anesthetic effects based on their solubility. However, that's not the case. These two compounds, F6 and trifluoromethylbenzene, are known to be non-anesthetics. You can give as much of it as you want to someone, they're not going to pass out. However, when we looked at the polarizability, what we noticed is that there's also a very nice curve here where the anesthetics fall along this nice line. However, these non-anesthetics do not fall in the same location, whereas all those other compounds fall relatively within the same location upon that curve. These other ones are lifted off of that curve. And so that gave rise to the idea that maybe polarizability does have something to do with the way that these anesthetics act. And so what we did is we performed a docking to find out where these anesthetics bind to microtubules. And then we calculate out the resonant frequencies of how all of the aromatic amino acids within tubulin oscillate back and forth. Now, you can imagine that there's somewhere up around 85, 86 of these aromatic amino acids, and they can either all oscillate back and forth at the same time, or some of them can oscillate one way while one oscillates the other way. And these all give rise to various oscillation modes. And these oscillation modes, obviously, they oscillate with different frequencies. And so we calculated that out, and that's what's shown on this bottom graph here. So that's the frequency in terahertz, and those light gray lines are the natural frequency modes of these aromatic amino acids in tubulin. Now, when you take an anesthetic and put it in there, it does two things. One is because you're adding an additional oscillator within the system, you get an additional oscillation mode, and that's those colored lines, and they're labeled there by the various anesthetics that we were looking at. as well as they slightly, very slightly on this scale, change the natural oscillation mode of tubulin. But you can't see it here because this is a terahertz scale, and those changes in oscillation are on the gigahertz scale. And so one thing that we notice is that the non-anesthetics, F6 and trifluoromethylbenzene, are on the left hand of this spectrum and within the oscillation modes of tubulin, whereas the anesthetics themselves, starting at around just above 620 or so terahertz, is that those anesthetic, just above 614 there, you can see methoxyfluorane there, is that way, and going up is where you start to get these anesthetics, and they introduce higher oscillation modes within the system. Now, if we look at those individual changes in the natural oscillations of tubulin, we find a common trend such that each of the anesthetics themselves introduce a downward shift, a gigahertz shift, at about 610 to 614 terahertz. And you can see that same trend going down, but it's the shift itself is dependent upon the chemical in question. Now, if you look at things like trifluoromethylbenzene and F6, you find that at 610, trifluoromethylbenzene actually shifts it up, whereas F6 does not have a shift at 614. It does a little bit further down, somewhere below 600. But if we look at that point exactly where all the anesthetics seem to have the same shift downwards at around 614 and plot that against minimal alveolar concentration, we get this curve here. And when we do that, what we see here is that we have this curve here that fits nicely and explains where we have anesthetic taking place, as well as it correctly predicts where the non-anesthetics would occur on this curve, such that you would have to have an unphysical amount of pressure of these non-anesthetics in order to induce some sort of anesthetic effect. Now, this is theory. This is an idea. It's 100% consistent with what Stuart's suggesting, but it needs something. This actually needs to be tested within an experimental setup to see if this is something we can alter or modify in the real world as opposed to in a computational setup. Now, coming back around to what we were talking about with psychedelics, is quantum mobility theory would suggest that anesthetics and hallucinogens would fall on opposite extremes. So if we have anesthetics that are downshifting at 610 gigahertz and adding a normal mode that's increased in frequency, we would expect to have an opposite effect for things like psychedelics. We would expect a downward shift in the normal mode, in the new mode that's being added, as well as potentially an upward shift in that 610 oscillation frequency. So that's what we do. We decided, let's quickly look at that. I calculated this out about a week and a half ago, so it's very new. We have not yet analyzed the individual changes in the natural frequencies of psychedelics on tubulin. However, the new normal mode that's introduced into the system is far down in frequency compared to the tubulin frequency. So we're looking at around 350 terahertz, whereas the anesthetics were above 600, and the tubulin falls around that 500 upwards of 700 terahertz region. So this preliminary analysis is not inconsistent with quantum mobility theory, and it's something we're again exploring a little bit further. So in closing, microtubules are key structures capable of modulating synaptic plasticity, as well as, I didn't get into it, they anchor ion channels within the membrane, and so effects on microtubules can affect ion channel function. Microtubules bind and are influenced by certain microtubule targeting agents. In specific, today we were talking about psychedelic phenylethylamines and volatile anesthetics, which are two specific classes within the larger groups of general anesthetics and psychedelics. This binding occurs within hydrophobic pockets around polarizable amino acids. The binding alters the normal modes of these oscillating of these aromatics. It also alters the polymerization dynamics of these tubulin polymerizing into microtubules. Phenylethylamines add a lower frequency mode while anesthetics at a higher frequency mode. And these changes in oscillations may, in some way, affect the dynamics of how microtubules how tubulin comes together to form microtubules. We made a not a guess. That's not the right word. With speculation about how this may work, and it was that if you add an anesthetic to one of them, you're going to put these proteins to be off-resonant. And there's work by Marco Bettini's group is that if you have resonant proteins within a cell, they find each other much easier than if you have two proteins that are off resonance. And so these anesthetics may be producing off-resonant behaviors, which would prevent them from coming together. We don't know what's happening with the psychedelics at this point. That all being said, further analysis is warranted. We're seeking funding for this. If anyone in the audience is looking to spend some money, come talk to me afterwards. Otherwise, I'll keep you posted as the years go on here. So here's the references. Take a quick snapshot if you want them. I'm not sure if they're posting these presentations. Otherwise, if you have any other questions, reach out to me and I'm happy to answer them. Thank you.